Hello, my name is Erone. I am a high school chemistry teacher at Rodriguez High School in Fairfield, and here I'd like to show you the results of my summer research experience with the Joint Genome Institute and microbial genomics. The context for this research is oil. As you may already know, oil has its many uses: gasoline, other fuels, other petroleum products, plastics. There are many uses, and the United States has been the leading producer and consumer of oil in the recent years. However, as you might also know, consuming all this oil, burning it, does have its detriments. Emissions are the leading cause of climate change, and one of those effects of climate change are these increased global temperatures. The production extraction process of this oil is also not as clean. What you're seeing here is the Deepwater Horizon oil spill of 2010, and 11 years later, we can still see the environmental damage that has caused. And so this does lead for a call for change. Now, rather than have to invent something from scratch, this approach looks at microbes and the biological processes that already exist. Rather than look at one microbe, one genome, this is a metagenomic approach, which looks at the DNA of a given environment. So what you're seeing here is that a sample is obtained from the environment, and the total DNA is then sequenced and reconstructed. The main benefit of doing this is that you skip the lab cultivation step. And so studying these microbes has been limited by how we know to cultivate them in the lab. So for instance, our knowledge of anaerobic microbes that don't need oxygen has been limited by the technology and skills developed to cultivate them in the lab. And so all of this genomic data and metagenomic data, this is made public and useful by the Joint Genome Institute. Their goal is to make all of this public for people to conduct their research, specifically with focusing on bioenergy and these microbiome processes. All of this genome metagenome data is available in the Integrated Microbial Genomes System, or IMG, complete with analytical and visualization tools, and that was my main method of research over the summer. And so tying this back to oil, I used IMG to answer these two questions. Seeing if oil degradation pathways played a role in the bioremediation of the Deepwater Horizon oil spill, and assessing this activity across all known and unknown microbes. And secondly, looking at a specific alkane synthesis pathway as a potential alternative to drilling for oil. Now, firstly, dealing with the Deepwater Horizon oil spill, this specific research dealt with anaerobic oil degradation. Anaerobic for two reasons. One, anaerobic conditions are hard to recreate in the lab, and therefore not much research has been done regarding anaerobic conditions. And secondly, anaerobic conditions sort of matches what happened with the Deepwater Horizon oil spill. Much of the research efforts was keeping the oil near the ocean floor where there is no oxygen. And so it's worth asking if the oil is still there or has something happened to it over the past years. And to do so, I found these anaerobic biomarkers, and I qualitatively assessed them across the Deepwater Horizon oil spill samples that are available in IMG. You are seeing the results of this qualitative analysis here. As you can see, the coastline sediment samples on the ocean floor, these biomarkers are detected there. And the plume samples where there is air mixing, totally fine that there are no results here. And so these biomarkers were also assessed across all known isolates and uncultivated metagenomes. And you're seeing the results tabulated here. As you can see, while there is some knowledge as to where these biomarkers are, there are still these unknowns that have yet to be fully sequenced, have yet to be discovered. This delta proteobacteria, these are known to use sulfur and sulfate compounds in their metabolisms, but even then, there are still novel Delta Proteobacteria waiting to be discovered. You're seeing the results here. And so with regards to oil and oil degradation, anaerobic oil degradation has been confirmed at the Deepwater Horizon oil spill sites, and there is still a lot to discover when it comes to microbes that have this function. Although I looked at just the Deepwater Horizon oil spill, there are other oil spill sites. There are other locations where microbes would have access to oil, such as tailing ponds. And so it could be worth bringing this method of research into those locations as well. Now, this whole part of the research was about bioremediation, fixing the damage that has been done. If we're looking for a true alternative to drilling for oil, the best way to prevent future oil spills is to not have to drill for oil in the first place. And so this does lead into the second part of this research project. And so there are microbes that are known to produce oil. In this case, cyanobacteria can produce them photosynthetically. Now, the pathway is known, how the cyanobacteria do this, but the genes at the time were not. And I say at the time because this research has already been conducted in the lab, but the focus for this part was using the tools available in IMG in order to tell this story. And so what was done was that individual cyanobacteria strains, their genomes were sequenced and they were in the lab tested for their alkane synthesis. There were strains that produced alkanes and some that did not. In order to find the genes responsible for this alkane synthesis, I performed a subtractive genome analysis in IMG. What this is essentially doing is that it's computing any genetic similarities across all known alkane synthesizers, and it removes the similarities from all strains that do not produce these alkanes. And so you are seeing the setup in IMG. You're also seeing a portion of the results. Now, IMG also has a set of visualization tools. And so one of those tools is the gene neighborhoods, and you can see how the genes are actually mapped along a DNA strand. What I'm showing here is that there are two genes of interest, and IMG, the gene neighborhoods, can actually show you that they're adjacent to each other. This heavily implies that they are involved in a related dysfunction. These are two steps that serve a related purpose. And this was confirmed in the lab. You can see the confirmed pathway from their experimentation. Specifically, these genes were extracted and placed in E. coli, and they were co-expressed to confirm alkane synthesis via this two-step process. Now, rather than look at a few isolates, if you want to look at all cyanobacteria, IMG can also help you search for this function across all fully sequenced cyanobacteria. What you're seeing here is the results of that function search. At the time of writing, most of the cyanobacteria, 185 out of the 203 genomes available, are indicated to express this alkane synthesis function. And so for research, this does inform you as to what you can do in the lab now. If you are interested in the slight differences in their enzymatic pathways, if you're interested in their environments, pH, salinity, temperature, amount of sunlight, IMG has that information available. And you can then pursue the ultimate goal of trying to find the engineer a strain of cyanobacteria for maximum alkane yield. However, I'm technically not a scientist. I'm a chemistry teacher, so my goal, my efforts are more interested in bringing this into the classroom. And so, does IMG have a place in the classroom, in the science classroom? Simple answer is yes. There is a reliance on setting up the hook, setting up the context. Full disclosure, all of this is currently a work in progress for the upcoming school year. And so part of my research efforts over the summer dealt with hydrocarbon oil degrading bacteria and hydrocarbon producing bacteria. This does lead into the notion of a short-term hydrocarbon cycle. So in the absence of oil spills and tailing ponds and other oil sources, these oil degrading bacteria have to exist somehow. And these cyanobacteria can act as a food source with the alkanes that they produce. And so currently in the works is a lesson series that introduces this. And the goal is to teach students basic chemical reactions, products, reactants. Amounts can be brought in as well, and that leads into stoichiometry. And this also happens to touch upon some of the earth science standards with energy and climate change. However, I do also concede that talking about DNA and genomics is perhaps more apt for life science classes. And so I also did spend some time postulating some scenarios where a biology class or an agricultural chemistry class can use IMG in their classrooms. And these are the scenarios which I will be sharing with my fellow science colleagues once the school year starts. And with that, I would first like to thank everyone involved with this research experience, especially everyone at JGI for their knowledge, their support, and their patience as I learn everything from the ground up. I had zero microbiology experience going into this, and so thank you very much for all of your support. And I do have this code here. If you would like to see the step-by-step guide to the alkane synthesis analysis at IMG, the code is provided here, aimed at beginner users like myself. And that's all I have. Thanks for listening.